Thank you very much, Neil. Very kind introduction. And yeah, as I say, as Neil said, my name's David Church. I'm a medical oncologist in Oxford. I run a lab in Oxford and we work on bowel cancer and womb cancer. We do research on genetics of that and hence the interest in Lynch syndrome. And today I'm going to talk to you about our efforts to work up a vaccine for Lynch syndrome and also a bit of background to the area and some other efforts from others in this space, of course. So I should also mention that I lead Kevin and Adam's transformation project for Central and South, which is making good progress, I think. So kudos to them, of course. So first of all, I just wanted to highlight the challenge that we face as humans or as any living organisms in copying our DNA, which is really quite exceptional, actually. So this, in schematic form, on the left is a human colon. Every day in your colon, about 10 billion cells die, and they need to be replaced. So truly mind-boggling numbers of cells that are replaced, and that's going on all the time, unbeknownst to all of us, of course. So here we have a little schematic of the intestinal crypts, the colonic crypts, and you see here the cells at the top of the crypt here. At the top of the villus is the structure that we refer to. They're red there, they're dying, and they're being replaced by these cells proliferating in green at the bottom. And for every one of those 10 billion cells, they need to copy all of their DNA before they divide. And copying all of their DNA means they need to copy 3 billion DNA bases, as we call them, little units of the spelling of DNA. So that all needs to be copied accurately as well. And that process goes on and on and on, unbeknownst to any of us, of course. And it really is very, very accurate. And why it has to be accurate, because if it doesn't, you have something called a mutation, which is an error in your DNA. And if that's an important part of your DNA, that can cause a defect in a protein, and if that protein regulates something like how quickly your cells grow or how likely they are to die, when you have a certain number of those, then you can get a cancer. And of course, we all know about the cancer risk with Lynch syndrome, we heard very nicely earlier on this morning. So how do we manage to prevent mutations from occurring in our DNA when this is going on all the time? The answer is that the machinery for copying our DNA is exceptionally accurate. So first of all, we have the machinery which actually copies the DNA, something called polymerases. They have some special features which mean that they're exceptionally accurate. And what they do is, first of all, they're very, very good at correctly pairing these letters in our DNA with each other. So their error rate in pairing these letters as our DNA is copied is about 1 in a million. And then they've got a little bit highlighted in pink and magenta there in these proteins, which double checks that it's been correctly paired. And if it hasn't, it stops, backtracks, and removes it and corrects and puts the correct one in, puts the correct base in, as we say. And that reduces the rate of error down to 1 in 100 million, as you see there. And after that, any errors that escape correction by those processes, which are already exceptionally accurate, are surveyed by something called the mismatch repair system. Now, this will be familiar to all of you, as many of you have Lynch syndrome, of course. As the molecular defect, the underlying cause of Lynch syndrome is born with a faulty copy of a mismatch repair gene, as we've heard from Fiona earlier. And what the mismatch repair system does is it scoots up and down the newly replicated DNA, and it checks if there's any errors. And if there are, it's able to recruit machinery to come in and repair them. And that takes the rate of error down to 1 in about 3 billion. So actually, every time a cell divides, the error rate in its DNA is about 1 per cell division. So it's exceptionally accurate. And that's why we don't get cancer at vastly higher frequencies that we do as humans. And as I mentioned, with Lynch syndrome, you're born with one faulty copy of a mismatch repair gene. And as a consequence of that, if you lose the other functioning copy of the gene, it increases the risk of mutations. And that goes from that 1 in 3 billion up by about 250-fold, we think, from studies in yeast, up to maybe 1 in 100 million or thereabouts. So a big increase in the risk of mutations, or big increase in the rate of mutations, and consequently bigger risk of getting defects in proteins and bigger risk of cancer. And this is my very impressive animations you're about to see, so please bear with me, they may well go wrong. But mutations make abnormal proteins. So about 1% of our DNA makes protein, and if you have a mutation in that 1% of your DNA, it can make an abnormal protein depending on where it is. And if it's affecting something that regulates how quickly cells grow or divide, then obviously that can contribute to cancer. But what it can also do, so this is normal structure of a DNA here with functioning mismatch repair, and here you have something called bases, as we mentioned, and they're read in 3. So every 3 bases makes 1 amino acid, and then the amino acids in the chain make the protein. So if you think about that, it's like a translating the DNA code into protein in 3s. And what happens is that you have amino acids, one after the other, and eventually when it gets to a particular combination of 3 amino acids, it tells the machinery to stop, and then the protein stops. And that's how we normally replicate proteins and make the proteins in our body, which is going on all the time. But if you lose mismatch repair, then, as I mentioned, the machinery loses the ability to recognize that there's errors. So here we see that these little 2 bases there have come off because the machinery for replicating, for copying the DNA is not perfectly accurate, as we've said. And so those two bases are lost. And then you have this slippage here, which is what microsatellite instability is. So now you see here the sequence of the DNA has changed. And because it's read in 3s, that means that the protein is no longer stopped at that particular point. And instead, it carries on. So the protein carries on. This is an abnormal protein, which may contribute to cancer depending on where it is, of course. So this is a new protein, which didn't previously exist in that body. And that's important. It's important, as I say, because if a mutation is an important gene, that can contribute to cancer, but it's also important because it can regulate the immune response to that abnormal protein. And immune cells are always being shown proteins in our body. So here we have a very simple schematic of a normal protein being shown at the surface of a cell by something called the MHC molecule, or human leukocyte antigen in humans. And it's shown to the immune cells. And if the immune cell recognizes that the protein is normal, so it's present in your body under normal circumstances, it's happy with that and it doesn't do anything. But if the immune system is shown an abnormal protein, it recognizes that and it says, hey, you're not me, and it goes after it. So the immune system gets angry. And this is how we fight viruses and bacterial infections. So these abnormal proteins from the viruses are shown on the surface of our cells, and then they're recognized and killed by the immune system. So the immune cell comes in, and it's able to kill the cancer, showing the abnormal protein to it. But cancers, as we know, are sneaky. And so what they do is they can regulate, they can switch on proteins on the surface of the cell and induce proteins on the surface of the immune cells, which are able to damp down the immune response against them. So they're able to silence the immune response against them. And that's how cancers with abnormal proteins are able to grow. They make these immune cells exhausted, as we say. And that's important for me as a medical oncologist because that's what we target with immunotherapy. So immunotherapy drugs go after those proteins, PD-1, PD-L1, as you see there, and they're able to reactivate the immune response against the cells having these abnormal proteins, and then the immune response is able to kill them. So it's turbocharging the immune response against the cancer, switching off, removing that invisibility cloak, if you will, that the cancer has. And what vaccination does is it boosts the immune response. So what you do with vaccination when you have your childhood jabs, you're shown a protein that you haven't seen before that's from an infection. And then your immune cells are able to recognize that and have a turbocharged response if you get that infection. And it's the same principle with cancer. So what the aim of the vaccination is to show the immune cells an abnormal protein, so you give it a heads up, if you will, that it might be exposed to that. And then what you do is you make the immune response turbocharged against that. And then if it occurs, then the immune cell is able to come in and kill the cancer cell that has that abnormal protein. So it's killed before it gets a chance to grow and become a pre-cancer or a cancer, where it's switched on that invisibility cloak and it's able to circumvent, escape from the immune response. So you're getting in there first and preventing it from developing those escape mechanisms, if that makes sense. And why is Lynch syndrome relevant here? Because it offers us a lot of targets for vaccination. So we talked about microsatellite there, so this repeat of the same base, the same letter in your DNA. And these are important in Lynch syndrome because these are the bits where the copying machinery that copies our DNA sometimes slips. It doesn't do it accurately. As a consequence of that, you get those little deletions or insertions, and they cause the mutations that can lead to cancer. But they're throughout our DNA. There's hundreds and hundreds of thousands of them. And even in the bit of the 1% of our DNA which makes protein, there's 400,000 of these little microsatellites, which are all prone to slippage and mutations and making abnormal proteins. And here we have a study from a group in the US from a few years ago, analysing cancers from a big cancer profiling effort called the Cancer Genome Atlas. And what we're seeing here is the frequency of alterations of abnormal proteins in proteins across the DNA in cancers with mismatch repair deficiency. Now, not all of these are from people with Lynch syndrome, but the molecular defect is the same. And what you see here is that looking at the numbers that we have, bowel cancer, 45 cases of bowel cancer, 64 cases of stomach cancer, 75 cases of womb cancer. And if you look at the numbers on the left, on the y-axis there, you see that these genes, which are mutated, leading to abnormal proteins, are very commonly and recurrently mutated in people with these mismatch repair deficient cancers. So you get predictable alterations, if that makes sense. And that's important because if you make a vaccine against those abnormal proteins, those predictable alterations and those predictable abnormal proteins, you can prevent, or you hope prevent, people from getting cancer. And this is maybe the furthest forward in the clinic. This is a Swiss company, Newscom. And they used cancers from the Cancer Genome Atlas, which is this big cancer profiling effort done in the US. And they looked at these recurrent alterations in DNA and protein that are present in cancers with mismatch repair deficiency. And they looked at all of those that are present in more than 5% of cases. And they identified several hundred of these alterations. And they've made a vaccine against 209 of them. So these are 209 abnormal proteins that are caused by loss of mismatch repair. So the molecular defect that if you have Lynch syndrome, your cancer has. So far, this vaccine has been tested in combination with immunotherapy, so with the drugs that target those invisibility cloak proteins that I showed you a moment ago in people with advanced bowel cancer. They've been found to be safe. And at the moment, recruiting in the US, they're recruiting people with Lynch syndrome at the MD Anderson in Texas and a few other sites, not currently in the UK, testing whether these vaccines are safe and testing whether they have any evidence of activity in people with Lynch syndrome. We're doing something along this lines in Oxford, which is our LynchVax project. So this is a project funded by the University of Oxford and Cancer Research UK. And we're using, rather than this cancer profiling effort that was done in the US, the Cancer Genome Atlas as it's called, we're using a different set of data that we've generated in the UK and England through the Genomics England project, which was an effort to sequence 100,000 DNA sequences from people with rare diseases and cancer. And we've used that to define our recurrent targets. And we're also studying pre-cancers as we want to prevent tumours. So how are we doing so far? So these are our cases, our cancers for defining these recurrent alterations. So you see here on the left, we have 2,000 bowel cancers, 2,000 colorectal cancers, and 665 endometrial cancers, womb cancers. And because mismatch repair deficiency is common in both of these cancer types, we have very high numbers of mismatch repair deficient tumours. So you remember a study I showed you before had 45 and 75. Here we've got far bigger numbers. So you've got 360 bowel cancers with mismatch repair deficiency and 202 womb cancers with mismatch repair deficiency. Now, most of these are not due to Lynch syndrome, but the expected fraction are. We know that from the testing that we've got. And here we see, looking at these recurrent predictable alterations, there are very, very many of them. So there are some advantages of this data over what's been used by others, actually, which I'd be happy to talk about at lunch. And here we see the number of unique alterations present in more than a quarter of the bowel cancers with this mismatch repair deficiency and the womb cancers. And you see intersecting them here, there's more of those in bowel cancers than there are in womb cancers, but if you put them together, we found 29 that are present in more than a quarter of cancers with this alteration for both types. So it's not just looking at bowel cancer, we're also looking at womb cancer, because obviously that's important. Getting some of the other cancer types is a little bit more challenging, as I'm sure you're aware. We've then looked at, speaking to your thing about biopsies, what do you do with them? Well, this is what we do with them. So we've looked at polyps, so precancers from the colons of 16 people with Lynch syndrome, 19 polyps. And what we see here is we've done, again, sequencing of the DNA to get all of the DNA sequence. And here you see the number of alterations in that person's DNA, in the polyp's DNA. And what you see here, those high bars there, have a very high number of errors. So not all polyps in people with Lynch syndrome have a very high number of DNA errors. They're not all microsatellite unstable, mismatch repair deficient, but a decent fraction are, about a third of them are. And you see already they have a very high number of mutations. And what's important here is that they have a very high number. They already have a good number of these recurrent predictable alterations as well. So there's 29 we're looking for, and we see between 10 and 17 already in these very, very small precancers in people with Lynch syndrome. So they're there early, which is good from the vaccine point of view. So what are we doing next? So we need to analyse more samples, including precancers of the womb, which so far we haven't sampled. We need to test whether the immune response against these targets is encouraging. We need to prioritise them to design a vaccine. And importantly, and something to be really, really keen to interact with everyone in this room about, is we need to get the opinions of the people we want to help, so people with Lynch syndrome. So what's your opinion on a vaccine? Should it prevent all cancers? Some more important, and what would concern you about vaccines? Because obviously, as a healthcare professional, as a doctor, I would say we've got an effective vaccine for COVID-19. It works very well. It saves lives, but not everyone wanted a vaccine. So there are obviously concerns about vaccines, many of which are justified, of course. And so that's what we've done, and that's what we're doing. So to sum up, scientific progress and our improved understanding of cancer means that vaccination for Lynch syndrome is on the way. We're not there yet, at least Newscom is recruiting, but we need more vaccines than just one, of course. At the moment, I think it's fair to say it's unclear what the best targets or the best approach is. There's different vaccine strategies, different targets, of course. Our Oxford Lynch Vac project aims to define this. And as I say, a proven vaccine is a little way off, but we'll keep working on it and keep you updated, of course, hopefully at a future meeting. So big group that we have here. So Francois Heil is our project manager and does a sterling effort. And I'd just like to highlight the really important contributions of our PPI group. So that's including Tom, Helen and Anna, who are here today. So thank you. And a big effort. So this kind of science is very collaborative. It takes lots of people, of course, and grateful to everyone for that. So as someone with small children, obviously all those smiling faces could only be generated by AI. Very happy to, I hope that's of interest, and very happy to take any questions, of course.
Just to come back on the Newscom vaccine, so it was invented by Roberto Cortese, who's the late leader of that team, and he, in fact, originally made it to do the Ebola vaccine. So we know it's a safe combination of viruses. So it uses a monkey adenovirus and a variant vaccinia, the old cowpox vaccine. And the nice thing is it was given to thousands of people in Africa for the Ebola vaccine without any major problems. So that gave it a head start. And so the idea of putting the frame shifts in, we've been working on for a long time. One of the anxieties was there might be an overreaction if there were too many frame shift markers in. But that seems not to have been the case from the American testing. But the problem now is that, of course, we've all lived through COVID. We've now all got a whole new generation of RNA vaccines. So the company are struggling a little bit because they feel like they're an old-fashioned vaccine. So we're not sure that they're going to be able to get enough money to get it out into full trial. And that's their biggest challenge at the moment. We obviously hope they can. But obviously what David and the crew are doing is coming at it from first principles. So it may well be that that actually overtakes the Newscom vaccine, just because the investment community are so married to RNA vaccines now that it might be hard to keep the old style religion working.
But he's got another 3 years, you know. If
Sure.
he waited in this country, he'd have to wait 3 years. So he's got
Well, but what they if it works, if it works there, that's great. They're doing the trial, and obviously we can adopt it at pace if they get some evidence that it works. All we know now is that it doesn't. Yeah. Well, it's unlikely they're going to prove efficacy in that short a time. There's not enough cancers going to happen in that small number of people. What they're trying to establish is it's safe to use, and then we have to follow for a lot longer to find out whether it actually prevents cancers. Yeah.